Good morning, Dr. Manoj. Myself, Dr. Suman Ray from NISpR. You are mute, sir.
Now I am audible.
Yes. You're
Good
audible,
morning, sir.
Good morning everyone. Good morning.
Other than the guest and whoever has to be on the dais, please switch off your videos also.
Akshay.
Good morning, sir.
Good morning. Good morning.
Sir, good morning. You are mute, sir. Somu, sir.
Parmanand has joined.
No, I didn't see
Yes,
him.
ma'am.
Okay.
Acha.
Yeah, good morning.
Okay.
Good morning, sir.
Good morning. Very good morning.
I think we should wait for some 2-3 more minutes.
Yeah.
Other people have to join.
Yeah, we can wait.
Yeah. And Pawanji is ready with the live streaming?
Pawan.
Okay. Good morning, Pawanji. Oh, okay.
Actually, this millets, it's quite. When Charu came 3 days ago and said about the topic, I really got, that kind of interest. See, basically, I'm from a village background. Okay, so I know what is the importance of this. Indigenous grains.
to commemorate World Food Day under NIScPR-SVASTik lecture series. World Food Day is celebrated on October 16 every year with the aim of shedding light on the millions of people globally who lack the means to afford a nutritious diet and the crucial necessity of consistent access to wholesome food. Dear all, we are honored and pleased to have with us Dr. Manoj Prasad as the keynote speaker of the day. He'll be talking on feeding the 10 billion by 2050, a genomics perspective. Before I hand over to him, let me briefly introduce about him. Dr. Manoj Prasad is working as a professor and J.C. Bose National Fellow at DBT, National Institute of Plant Genome Research, NIPGR, New Delhi, India. He did his BSc in Botany Honours, MSc and PhD in Botany from University of Calcutta. Prior to working at NIPGR, Dr. Prasad served as Alexander von Humboldt Fellow in 2002-2003 at the Leibniz Institute of Plant Genetics and Crop Plant Research, IPK, in Germany. His research interests include plant molecular genetics, genomics, and stress biology. Dr. Prasad has published more than 200 research papers, reviews, and popular articles in highly reputed international journals like PNAS, Plant Journal, Journal of Experimental Botany, DNA Research, Cellular and Molecular Life Sciences, and so on. He is working and serving as associate editor in renowned international journals of high repute. He is the recipient of many prestigious awards and honors, to name some of them. Fellow of all the three national academies, Silver Jubilee Commemorable Medal in 2022, Indian National Science Academy, New Delhi, J.C. Bose Fellow, and in 2018, Department of Science and Technology, Government of India, Tata Innovation Fellow, 2017, Department of Biotechnology, National Bioscience Award, 2015, Department of Biotechnology, Government of India, Birbal Saini Medal, 2021 of Indian Botanical Society, India, Alexander von Humboldt Fellow in 2000, Germany, Golden Peacock Award, 2019, and Hira Lal Chakrabarti Memorial Award 2010 to 11, Young Scientist Award, NAS, New Delhi in 2003 and 2004. Friends, we are very much pleased to have with us Dr. Manoj Prasad. And now I would like to, I welcome you, sir, on behalf of CSI NIScPR. Very warm welcome once again. I would like to hand over to Shri Jaisomu, sir, Chief Scientist from NIScPR, for the welcome and introductory remarks. Over to you, sir.
Yeah, thank you, Dr. Suman. It was a very wonderful introduction about our speaker today, Dr. Manoj Prasad, Professor and J.C. Bose National Fellow, National Institute of Plant Genome Research, New Delhi. Sir, it is actually it's a great pleasure to have you on this day. The event related to this World Food Day, and the team that is organizing, the SVASTik team, led by Charulata and Param. Actually, this is one of the top projects of our institute. See, when we say India, of course, we are rich in tradition, and all of us, talk about traditional knowledge, and there has been documentation of this traditional knowledge going on for the last 2 decades. And now, with the thoughts of Honorable PM, the project has come up. Why not we scientifically validate this existing knowledge and share with the people? And we have that one of the best team of us we have in this job. And until now, I think they have done about 42 stories, okay, and covering about 17 Indian languages, right? So, and this is the best platform and we welcome you to share your thoughts on the traditional food systems. Okay, I was just seeing your bio. See, in 30 years since 92, you have earned 24 awards. It's not a joke. Okay, and Suman was just listing one by one. And another thing, actually, see, we NIScPR, if publicly funded publishing institute, if we say, actually we rank the top one. Okay, we are at the top, and we have a legacy of 8 decades. Actually, way back, it started as 2 units. One, as of course, you must be aware of the great encyclopedia, Wealth of India. Okay, and another project was JSIR, Journal of Scientific and Industrial Research. These 2 units actually started in 1942 along with CSIR. So that is the base of us. And late in 71, this policy research came, and 14 we got merged. So now we are very youngest in the CSIR family, and we are very glad to have you today. And I was just had a glance on your topic, feeding the billion by 2050. Okay, I think, sir, you have to double it up. I mean, feeding the billion, actually 10 billion. I was just calculating the growth, Indian population growth for the last 2 years. It is 0.9. Okay, so if you calculate for another 22 years, by 2050 we will be almost 20 billion. So you have to develop that. Okay, so that was just this one. And this, I was seeing your publications, and you worked on foxtail millet, minor millets, right? And in Tamil, actually Tamil language, we have value for millets. Okay, and especially I was just looking into fox millet. English name I saw that actually fox millet, and Kangni maybe the this Indian, this one, and in Tamil we call Tinai. Okay, so in earlier text, old text, you can find this phrase, Thenum Tinai Mavum. Okay, Ten is honey, and Tinai is foxtail. Now I understand. The field here that you are working in. Okay, so we are very glad to have you, and we thank, and on behalf of CSIR and NIScPR, I say Jaya Somu, Chief Scientist, welcome you and share your thoughts. Over to you, sir. Thank you.
So first, I think I should share my slide actually. Huh?
Yeah.
So there's a share option.
Then
You just have to share your screen and then select your PPT if it's open in your desktop.
PPT is open, and then I share.
You share your screen. There are a lot of options will be there. You need to share your screen. You need to share your screen.
You can see that I have made it full screen.
Yes, sir.
Is there?
Yes, yes.
Now it's moving?
Yes, it's moving in.
Okay. So good morning, everyone. It is a nice meeting you all through this virtual platform on the occasion of NIScPR-SVASTIK lecture series to commemorate the World Food Day 2023. And my foremost thank is to Dr. Ranjana Agarwal, the director NIScPR, for giving me this opportunity. I would also like to thank Dr. Suman Ray for nicely introducing me, and Dr. Jaishamu for telling us the importance of millet. And I would also like to thank Dr. Pramananda and Charulata for conducting this Satvik lecture series. And I would also like to thank all the audience who are present online to listen to my lecture. So as you see that my talk I have listed as feeding the 10 billion by 2050, a genomics perspective. So I will give you a example how millet can play a major role in rescuing the food demand. So when I try to prepare the slide for today's lecture, I just try to find out what is the current world population, and I found that it is more than 8 billion. And it has been proposed that by 2050, the population will be 10 billion, maybe more actually. So now the question is, do we have enough food to feed these 10 billion people? The answer is no. So it has been told from our childhood that to live on this earth, we need 3 basic things. Number 1 is food. Without food, nobody can survive. Then comes the shelter and the cloth. So there's a famous movie in Hindi, Roti, Kapda and Makan. So this is based on this need of basic needs to survive on this earth. So there is a problem for this because food is not available to everybody. According to this FAO 2020 WHO report 2020, 1 in 10 among us are still hungry. 10 seconds, a child dies from hunger-related cause, and 1 in 4 of the world's children suffer from malnutrition. So these are very chronic problem. So we have to address this thing. Now, if you ask that, as I said, whether all the 8 billion receive their food for the 3 times a day, the answer is no. So this is the 2021 Global Hunger Index by severity, and this problem is major issue, serious issue is in South Asia, including some part of India also. If you see the Global Hunger Index 2021 data, in 2021, India was 101 and out of 125 countries. The recent data shows that the Global Hunger Index, India ranks 111th out of 125 countries. So even Pakistan and Sri Lanka, Nepal and Bangladesh, they are better on this Global Hunger Index over the India. So when we check the literature, we found that there are 8 billion population at present. And out of this 8 billion, 820 million lives on this chronic hunger related region. And out of the 820, 193 million, this consists of 55 countries were facing the acute food insecurity zone. And these are mostly the Southeast Asia and some part of India. And this is before COVID-19. And after COVID-19, this 193 million, 55 countries become 345 million, 82 countries. And more countries has been included in this Global Hunger Index. So the problem is very serious, and India's position was also not good if you see this data. So UN has developed 17 sustainable development goals. Out of these 17, if you see that I have make it the highlight, 1, 3, 8, 12, 13, 14, and 15. 7 actually, out of 17 were directly related to the food. So food is very important if you talk about the SDG goal of United Nations. Now, what is food actually? If we see the Cube Botanical Garden data, the largest herbarium and botanical garden in the world, so there are more than 11 lakh vascular plants actually present on this earth. And over the time, due to selection and artificial or selective domestication, 80,000 plants has been used for food, fodder, timber, medicine, and other commercial purposes. And out of this 80,000, mostly the wild one, underutilized one, they are neglected due to the introduction of the grass crop or the staple crop, rice, wheat, and maize. And these 3, rice, wheat, and maize, cater more than 50% of the world food. So see here, we have more than 11 lakh vascular plant on this earth. Over the year, we use 80,000. And at present, we are using 3 major cereals, rice, wheat, and maize, to cater our whole population. So we are ignoring the underutilized crop plants, which are much more beneficial than the nutritionally much more important as compared to the major cereal crops. Now the question again I ask, how to feed these 8 billion people with just 3 plus 12 crops? So as I said that you have seen that there is a huge imbalance between the current population 8 billion and the number of crops and animal species to feed the 8 billion people. So there is a need to mainstream the underutilized crop for food and nutritional security. And how to do this? And for that reason, next generation genomics tool for trait improvement in this underutilized crop species is required, because you know that most of the research has been done till date mostly in rice, wheat, and maize, but very less research has been done on millets or underutilized legumes and tubers. Now, what is neglected? Neglected means less research attention has been made for this minor underutilized crop. And what is underutilized? Marginally cultivated and used. Marginally cultivated means where the lands are not suitable for cultivation of major cereals. Now, what are the example of this underutilized crops? There are several tubers comes under this: legumes, leafy vegetables, and minor millets. If I talk about tubers, there are taro, arrowroot, Indian shoot, ganola, purple yam, air potato, and elephant food. They are highly nutritious tubers, are very less in use. What I would talk about the underutilized legumes? Their example are wing bean, jack bean, bambara groundnut, kidney bean, fava bean, lima bean, cowpea, azuki bean, velvet bean, rice bean, and horse gram. I can tell you that rice bean has been sequenced by NIPGR recently. So again, all these underutilized legumes are highly nutritious as far as the protein and nutrient is concerned, or vitamin is concerned. About the small and minor millets, there are 10. Example are finger millet, foxtail millet, proso millet, little millet, barnyard, teff, kodo millet, funio millet, job steer, guinea millet, and brown stalk millet. So these are the wonder crop, and I will tell you and with example of data how they are giving the nutrition to the whole world. So anciently, these traditional crops, this minor millets, legumes, and others, they are basically cultivated in the rain-fed areas or the semi-arid region. But the introduction of this cash crop, rice and wheat, after the Green Revolution, 1965 to 70, their production and cultivation was restricted to very marginal land. So if you see the introduction of these millets over the rice, according to a recent report, we have found that millets are already available almost 8000 BC, when rice came much later, around 4000 years ago. So these are the data from the sequencing data, and these are 100% reliable data that millets are already in use by ancient people, including our forefathers and our ancestors, and rice came much later as compared according to this data. So if I talk about the statewide production of millet, according to the ICAR, Indian Institute of Millet Research, Hyderabad data, Rajasthan and Maharashtra, these two states are top in producing area of millet production, and Tamil Nadu and Uttarakhand, these are the two top state in India. Their yield is better than any other states. So yield, it means a kilogram per hectare. So you can see the quinquennial mean area production yield of the millet in India. So this time, 1965 to 70. So this is the Green Revolution time, and this blue bar is indicate the area of millet cultivation. You can see before this Green Revolution time, 1961, the cultivation of millet is more actually. But the introduction of the cash crop, rice and wheat after the Green Revolution, the area of millet cultivation was reduced dramatically. This is up to 2020 data. So, but the yield was dramatically increased by developing some new varieties or improved varieties by the breeders. But the area is really as concerned. So what are the benefit of these small or minor millets? If you see where our article in Trends in Plant Science in 2021, we have summarized the this minor millet. They are much superior in terms of agroecological traits, nutritional quality, and potential ensure the immediate demand for food security. If you talk about finger millet, Erusina coruscana, it is rich in calcium and polyphenols. It is very good for the replacement of this calcium, the tablets actually. So because if you see that 300 times more as compared to rice and wheat, the calcium present in finger millet. Okay, and good food for the toddlers, the kids. Foxtail millet, the tenai and this kangni, they're high protein content and food for diabetic and poultry feed. Because you know all the millets are basically good for type 2 diabetic people because they possesses good resistance starch, and this good resistance starch actually helps the conversion of carbohydrate to glucose slow, and that's why body can readjust the glucose level and metabolism level so that the carbohydrate immediately cannot be converted to glucose, which is the case of rice and wheat. Top of this, all these minor millets, they are superior in water and nitrogen use efficiency. So they require very less water to grow, or mineral, or these fertilizers to grow. So they can grow anywhere in the marginal land where soil is not much fertile. There's less water is required, and you can see here. Nutritionally, they are 3 to 5 times more superior than rice and wheat. I will show you some data. So these are our data. What we have shown that when we compare these minor millets with rice, wheat, and sorghum in composition per 100 gram of grain, in terms of protein, fat, as crude fiber, energy, and vitamin, thiamine, and riboflavin, you can see in every cases they are 3 to 5 times superior than rice, wheat, and sorghum. So millets are rich in protein, fat, as, vitamin, etc. as compared to rice and wheat, 3 to 5 times. And then if you talk about the micronutrient, again they are 3 to 5 times superior as compared to rice, wheat in terms of calcium, potassium, magnesium, sulfur, zinc, iron, boron, magnesium, nickel. Now coming to the tradition and cultural aspect of millet in the context of India. So these are nothing new actually. If you see in India, the Yajurveda text, it is already talk about foxtail. This is known as Priyangava, and barnyard millet Anava, and black finger millet is Samaka. So these are already actually exist in Yajurveda, and indicating that minor millet consumption was very common, predating to Indian Bronze Age, 4500 BC. There is a historical aspect also. This Magnetas, when he was the first foreign envoy to Secular's dictator of General Alexander the Great to the court of Chandragupta Maurya at the 302 to 298 BC, who mentioned about the cultivation and consumption of millet in India. So you see here our Yajurveda and other the historians, they also advocated that millet is already in practice used by the our forefathers or ancient people. If you see the Kautilya Arthashastra, it was also mentioned about the culinary properties of kodo and foxtail millet. So this kodo is Kodava and foxtail is Priyangu, will increase 3 times the original quantity when it was cooked. Again, the grains will increase twice to the original quantity if they are moistened, and 2.5 times when they're soaked to sprouting condition. This has been written by this Kautilya in his Arthashastra. So see this, that also the traditional and cultural aspect of millet that already documented by the renowned people of our ancient India. So what are the health benefits of millet? Actually, if you compare the millet bread with the maida and atta bread, if you see that millet bread are gluten-free and high fiber. Very important. I have seen several people have allergy to gluten. But if somebody take this millet thing, there is no gluten. As I said, this is good for diabetic food because the presence of good resistance starch. So it helps the conversion of carbohydrate to glucose very slow due to presence of good resistance starch in the millet. Millets are mineral-rich food and antioxidant. As I said, that in finger millet, the calcium is 300 times more actually. So there is no need to take any pills or supplement for calcium. So you can take this millet once or twice in a daily routine food. You can avoid these pills, supplement pills for calcium. Multi-benefit food, as I said, that it has good for any vitamins, fiber, anything. Food for lifestyle disease, as I said, that is good for the diabetic people. Again, if you talk about socio-economic aspect of millet in the context of India, it's very actually already well documented actually in the literature. So mainstreaming millet cultivation can ensure livelihood security and economic empowerment of resource for poor farmers. But we have found millet can grow on poor soil. As I said, they can grow in semi-arid and marginal land with little or no external input. Nothing is required. No watering regularly, no fertilizer, nothing. Millet require low water supply and capable of growing under non-irrigated condition even in low rainfall region. But there's a hardy crop. It can extreme environmental condition. It can grow more than 40 degrees. It can grow happily, where rice cannot grow beyond 30 degrees. Millet can survive in high temperature. As I said, it can go up to more than 40 degrees. Even in climate change, millet can withstand the climate change, and that's why it is known as a climate resilient crop. Millets are C4 plants. I know that rice is C3, wheat is C3, but C4 plants, millets, all the millets are C4 plants. They have a high photosynthetic efficiency. So that is the reason they can withstand all the climatic disaster. So millets are now slowly growing the popularity. So recently I visited this southern part of India, and mostly I used to go to visit the Tamil Nadu actually. If you see that when I visited the one shop for my breakfast, I found that 6 different types of dosas and all are from millet actually, and they cost more. If you compare with the normal dosa, it is more than 106 rupees actually. So keeping in view this, you can see that now importance of this millet actually. So in terms of culinary, that I have shown you in the ancient India, and even Kautilya has also said about this, that it was widely used by the Indian. And even if I talk about the burned millet biryani, you see here now in Tamil Nadu also you can find biryani with all these millet dishes actually. And if you want to buy millet in supermarket, it have to give more money, 110 rupees per kg for burned millet or any millet actually. But rice you can get it in 30, 40, even in 50 rupees, but not any millet. And if you talk about these Bollywood movies before this Green Revolution, if you can see that nowadays we have seen all the Hindi movies start their dancing behind this brassica field, sarson ka khet. But earlier it was all millet field. You can see here this Mother India, this background is in the pearl millet. Even for Sole also. So these are some documentary evidence that before this Green Revolution, 1965-70, all the movies has been placed in the millet background or pictured in the millet backgrounds. Now, keeping in view this importance of this millet, actually, Government of India, through Gazette notification of 10th April 2018, termed all these 10 minor millets, as I said, this finger millet, proso millet, kodo millet, burned millet, as a nutri-cereals for production, consumption and trade point of view. So by Gazette notification, Government of India also realized the importance of this millet in the current scenario of population explosion and the food shortage. So millet is only a rescue to this current problem, both climate change and also the number of population and the nutrition point of view. And for this region, the Food and Agriculture Organization of United Nations has declared the current year 2023 as the International Year of Millets, and with this SDG taglines. So you just see a 2-minute video I can show you. So this is the short video to tell all of us the importance of millet. Now the question is, can millet address the hunger issue? In 2021, just after this COVID-19, myself and my student, President Muthu, tried to write an opinion article for Trends in Plant Science to tell, yes, millet can address the hunger issue. So this is the integrated map showing hunger hotspot spanning 55. Now it is now the country is now 88 actually in the after the COVID-19. So which that based on this population size, they are living in the food insecure population. Okay, so these are the 55 countries actually. Now it is 88. So what are the roadmap we have proposed in our Trends in Plant Science opinion article? We have to identify the millet species suitable for cultivation in the particular climatic zone and soil. So say for example, in Madurai district of Tamil Nadu, I know that barnyard millet was well grown because that climatic and soil condition is good for getting good yield for the barnyard millet. Similarly, we have to optimize the cultivation practice and training of farmers for millet cultivation. So we have to make a device because you know that these they are now modern farmers. So we have to tell them that you have to optimize the cultivation practice, and this soil and this climate is good for this type of minor millet. So like I have talked about this barnyard millet for Madurai district in terms of soil and climate and the yield potential. Then we have to implement the large scale production of millet species and optimization of downstream processing. This is very important. If you produce something in the field, but if your downstream processing is not good, then everything will be lost. And fourth one is the improvement of millet yield and quality. And for that, people like us as involved for genetic improvement of millet varieties using modern biotechnological approaches. Because you know that millets are very problematic due to this high quantity of phenolics compound. And if you use a dough of millet, any millet, and you keep it for 15, 20 and half an hour, you will get a rancid taste if you prepare the roti or chow or whatever it is, because due to presence of good high phenolic things actually. So phytic acid, very high phytic acid in all the millets. And then finally, the supply chain, actually devising appropriate strategy for post-harvest processing, storage, and supply chain. This is good, true for all this production of cereals and other communities. So if you want to know more about this, you can read our opinion article, Small Millet Enduring Food Security Amidst Pandemic, because we have written this thing during the pandemic 2020, published in Princeton. And now I will give you two examples and how the next generation genomics tool can use to improve the trait in these minor millet crops. So the first one example is the classical example for characterization of the dehydration responsible event binding protein by Charu, who is now scientist in NIScPR. And she got all the accolades, was present at that time. She was the INSA Nasi Medal for Young Scientist, and then Sir Women Excellence Award, and her thesis was also judged as ICR Jawaharlal Nehru Outstanding Doctoral Thesis. She also got the Inspire Faculty Award. So one of the best student from my lab, she is my second PhD student in my career, and she published an article in Journal of Experimental Botany where she identified an SNP in the DRAPE gene, and to distinguish the drought tolerant and sensitive cultivar. So what she did, she developed one allele specific marker which can distinguish the tolerant variety from this sensitive variety, so that it can be used as a diagnostic marker for screening this drought and sensitive and tolerant variety with simple PCR, polymerase chain reaction. No need to grow the plant to see whether they are tolerant or sensitive, but with the use of this simple allele specific marker at the seedling stage, at the 14-day stage, you can tell that which plant will be tolerant, which plant will become sensitive. Now coming to the global analysis of stress responsive gene families to characterize the more gene families, we have used this genome sequence of foxtail millet. So all the millets are polyploid except this foxtail millet. Foxtail millet is diploid and similar to rice as far as the genome size is concerned, 500 MB. So keeping in view the importance of this millet, the two group, the American group by Benedictson et al. And Chinese, the Beijing Institute group, Zhang et al., they published this foxtail millet genome sequence information. And by using this information, we have developed 4 open access databases so that people who are sitting anywhere in the world, they can utilize these resources, whatever we develop in DBT-NIPGR New Delhi. So we have developed more than thousands of genomic SSR markers, simple sequence repeat. We have developed almost 2,300 transcription factor database. And we have also developed a 355 microRNA database of foxtail millet and more than 30,000 transposable element-based marker database. The 4 open access database, they have been integrated with the comparative mapping so that people can also can use it for comparing with their data with rice, maize, and sorghum and brachypodium with foxtail millet. So coming to this identification of these water responsive genes by using this genome sequence data, we have identified 586 genes which are functionally annotated to response to water. I have talked about Charu's DRAPE work that she has developed one allele specific marker. And then I want to talk about the heat shock protein. Heat shock protein is also known as molecular chaperone. It's also present in animal. Also, we also have the heat shock protein. So this is the work of Roshan Kumar Singh, and recently 1 year back he has got the degree. And for this work, he has got, like Charu, he has got ICR Jawaharlal Nehru Award for Outstanding Doctoral Thesis, and also he got the assistant professor position at Calcutta for his wonderful work in science. So what are these heat shock proteins? They have played multiple roles actually, and they produce the stress defense through protein-protein interaction to the target protein. There are 5 different types of HSP: HSP 70, heat shock protein 90, heat shock protein 100, heat shock protein 60, and small heat shock protein. So there are 5 different types of heat shock protein. They have several functions. one of the main function is to produce defense against adverse condition. So when Rosen searched the literature, he didn't find anything. So he used this bioinformatic platform HMM profiling, and then he identified 113 heat shock protein gene in foxtail millet. And out of this, there are different categories, like small heat shock protein is 37, heat shock 60 is 20. So before going to the characterization of this gene, Charu has characterized this 200 expression of foxtail millet through agronomical and biochemical and physiological procedure, and he identified that IC4 and Prasad as tolerant and IC41 and Lepashi as a susceptible cultivar. And these 4 contrasting cultivar has been used widely in my lab for doing all the expression studies. So when Rosen did the expression studies of these 113 HSP genes in response to a 4 abiotic stress condition in the 2 contrasting cultivar IC4, which is tolerant, and IC41, which is susceptible, and red indicate the highly upregulated and green regulate the downregulation, and he identified that 37 HSP genes were comparatively differentially expressed in contrasting IC4 and 41, the tolerant and sensitive cultivar. And out of this 37, he identified one small heat shock protein, it is HSP-27, which were differentially expressed in all the 4 abiotic stress conditions, that is dehydration, heat, salinity, and cold. When we check the individual fold expression of this HSP-27, 35-fold upregulated in tolerant cultivar during heat stress, 21-fold in cold stress, 47-fold in salinity stress, and there is a reverse pattern in susceptible here, more than 7-fold in dehydration stress, not the tolerant one. So what is the reason for this high expression? So when Rosen checked the upstream of this HSP-27, he identified an heat shock element signature, and heat shock element is the binding site for any transcription factor, and so that it can regulate the expression of this HSP-27. So when we did perform this relative expression and co-expression, Rosen identified a heat shock factor A2E, which was binding to the promoter of this HSP-27 in their signature heat shock element and regulate the expression of this. To prove this, he silenced this HSF2AE, the transcription factor, and HSP-27 simultaneously by using the virus-induced gene silencing technique in foxtail millet. So this is basically transiently tell you that if you silence this gene, whether this gene will function the desired rate or not. So virus-induced gene silencing is one of the technique which tells you quickly the function of that particular gene. So when he silenced this heat shock factor 2AE, which is binding to the heat shock element of the HSP-27, the expression goes down. Similarly, HSP-27, when he silenced, the expression goes down. So it indirectly says that these 2 elements are responsible for controlling the expression of this HSP-27. The question is that whether in planta it is doing or not. So these are all different experiment. So through yeast 1 hybrid and the luciferase assay. Luciferase is in planta in the plant system also. He showed that yes, if you mutate this HSP-27, there is no interaction. But when it is normal condition, HSF2AE, the transcription factor, HSP-27, interaction is there by lightening. So heat shock factor 2AE directly regulate the expression of heat shock protein 27 in response to thermal tolerance or high temperature stress. Now to prove this, the HSP-27 is act as the prevent thermal aggregation, he used substrate protein HSP-27 citrate synthase and alcohol dehydrogenase as to prove its thermal aggregation activity. So you can see here, without HSP-27, the scattering is more over the time period. But with HSP-27, 150, 30, and 450, the scattering is less. What does it mean? It means that HSP-27 prevent thermal aggregation of the substrate protein citrate synthase and alcohol dehydrogenase. Now, when it tried to find out the whether this HSP27 protect the enzymatic activity. So what he did, so he heated this cited synthase 45 degree till 40 minutes, and then it put it in the room temperature 25 degree. You can see without HSP27, this you can see no addition, and this 150 millimolar GST enzyme activity goes down after coming to the room temperature. But with HSP 150 millimolar, it can regain up to 40% of its enzymatic activity. So it is the HSP27 prevent irreversible thermal inactivation. When he increased the millimolar concentration of HSP27 from 50 millimolar to 600 millimolar, it can regain up to 60% of its enzymatic activity. So see here, 150, 40%. When he increased to 600 millimolar, 60% enzyme activity can regain by HSP27. When we check the localization of this HSP27 in the plant, he found that through Western blot, he saw that HSP27 was a chloroplast localized molecular chaperone, not present in cytoplasm, not present in mitochondria. Only it is localized in the chloroplast of the plant. And then he tried to find out what are the interacting peptides of this HSP27 in the chloroplast of the plant. So he used this LCMS approach, the proteomics approach, and he identified there are 80 peptides that interact with this HSP27, which belongs to photosystem 2, photosystem 1, molecular chaperone, protein associated with PS1, 2, light scorpion, and chloroplast localized protein. When we checked the phylogeny of this HSP27, he found that HSP27 of oxygen only present in C4 panicoid species, not in C3 plant or not in any other dicot species. So this is a very specific HSP27 for C4 plants only, not in any other plants, not present in rice, wheat, any other plants. So to prove his stress tolerance role against this heat tolerant thermal stress, he overexpressed this fox-terminated HSP27 in the IR64 variety, high yielding variety of rice. And when we check the agronomic traits and overall yield, he found that all the transgenic rice lines, these are the 3 transgenic rice lines over this is the wild type where this gene was not present in rice. And these 3 rice line, they harbor the fox-terminal HSP27. So you can see in terms of height, number of tiller, number of grain per panicle, panicle length, panicle per plant, and grain yield. All the 3 transgenic rice lines perform better as compared to the wild type. So you know that current scenario of heat stress is very problematic. So rice grow 24 to 28 degree. This is the normal condition. And if you see the 10 years current rice season growing time, the average temperature is 40 plus, but rice grow up to 30 degree. So if the temperature is 40, then how could you get the yield? And this is according to IPCC, the 4.4 to 5 degrees centigrade rise by 2050. So it is very difficult. You can see here, the whole rice growing region, it is totally red actually, means temperature is more than 40. So we mimic the current natural population from 32 to 45. Now we have done this experiment with 50 degree, mimicking the natural heat stress condition in the controlled phytotone chamber and evaluated Rosson's transgenic rice lines. And you can see here, when we evaluated the 3 weeks rice seedlings during high temperature stress for 8 days, followed by recovery for the 7 days, he found that only 22% wild type are surviving. When we check the ROS activity, you can see the wild type show high ROS activity as compared to the other rice 3 transgenic rice line. In terms of lipid peroxidation, you can see the wild type has more, even for electrolytic leakage as compared to the wild type. So HSP27 improve high temperature stress tolerance in transgenic rice line. When he checked the photosynthetic parameter in terms of total chlorophyll, chlorophyll A, B, and net photosynthesis, in all the cases, rice transgenic 3 lines, they perform better as compared to the wild type. When we check the protein in terms of with these transgenic rice lines and the wild type, there is no change in transcript level. But at the protein level, he found that the transgenic rice line proteins, they are not degraded as compared to the wild type. So it also shows that HSP27 protect chloroplast proteome integrity during high temperature stress. And that is the reason why the transgenic rice line can survive in high temperature stress, because proteins is not going to degrade actually. When we check the yield loss, he found that there is a yield penalty of 70 to 80% in the wild type, but in transgenic rice line 35 to 40. So in nutshell, what I have can summarize my talk like this: HSP27, a unique panicoid group of procedures, is present only in millets, not in rice wheat. HSP27 is chloroplast localized molecular chaperone, not in cytoplasm or mitochondria. HSP27 protects several chloroplast protein during high temperature stress, as I showed you from the proteomics analysis. Transgenic rice line, that IS64 variety, overexpressing this foxtail millet HSP27, displayed improved tolerance to high temperature stress. So it is now in the paper we have produced 45, but we have done the experiment now 50 degree and maintains higher photosynthetic efficiency. And the transgenic rice line show reduced yield penalty 30 to 45%, while compared to wild type 70 to 75%. So HSP27 maintains chloroplast function and improved thermotolerance. And Roshan and myself have filed the patent in 2020. We are waiting for the granting. So this is the thing that we have published this work in Cellular Molecular Life Sciences. This is the Society Journal from Springer. Impact factor is more than 8. And this is a transdisciplinary journal. So mostly the animal people they publish here, but we also try to publish in, because HSP is common for both an animal and plant. So what in nutshell I can tell you that millets are the wonder crop in terms of abiotic stress is concerned. So we have used that multi-omics technique. I have shown you a genome sequencing data, genome-wide characterization of this gene family, 113 we have identified. We have done the transcriptome, rice transcription. We have done the bioinformatics. We have done the metabolomics and proteomic analysis for the transgenic rice lines to find out what are the difference in terms of the wild type rice and the transgenic rice line. So we have identified the HSP27 through this integrated approach, and we have characterized this HSP27 through biochemically, biophysically, and through overexpression study. And we have developed a climate smart cereal crop of IS64, which can tolerate the high temperature, drought, and salinity. I have not shown the drought data, but we have also conducted the drought thing, because drought and temperature stress, they are correlated. So we have developed the climate. So in the long-term goal, as I said, how to fit 10 billion by 2050, design the cereal crops which are more durable in adverse climatic condition amidst the threat of global climate change to ensure food security for 10 billion people. Now coming to this, we have published several papers on different aspects. And then the success story of my student Charu, I have talked about this Roshan. Muthu actually, he also got like Charu, all the things. So he also characterized this, developed these open access databases, and also characterized several gene families. And Swati has characterized the NAC transcription factor. And these three guys, they worked on my plant virus interaction. So what is the reason for the student success? The thing is this, what I have given, that 100% independence to experiment on their own, no stringent evaluation and performance except one monthly lab meeting, because you know that research is a very actually painful thing. It's not an easy thing. So I generally try to give less pressure to the students so that they can give some space and they can freely, they can think freely. Special attention towards encouraging writing and speaking, because these two skills are individual skill. Nobody can teach anybody how to write and how to speak. So individual skill they have to develop. Exposure to editorial process like reviewing and editing. I gave all my postdoc, not the PhD, but the postdoc to review the papers and the thesis so that we can sit together and we can improve it. Freedom to attend any conference, symposium, and present their work. This is very important, because you know that if you don't interact with people, you don't know to how people speak and what is going on in the world. So that is many things you can learn from attending symposium and conferences. liberty to apply for a fellowship award and whenever eligible. This is very important. Until unless you apply, how do you know that you will get it or not? Students are encouraged to visit other labs to get assistance if required. This is also very important because no lab is full sufficient to do all the experiment. So we need help from each and others. Students also encouraged to assist the other lab if they helps out. This is also very important. This is not a one-way traffic, it's a both-way traffic. So we have to help each other. What students should keep in mind, they should have the curiosity and persuasion. Because I tell all my students, the PhD is not for everybody, bread and butter. So if you have curiosity and persuasion, if you have a strong desire to learn something new, if you have loved the subject and patient, open-minded and cross-border thinking, ability to ask questions, if you have these 5 ability, then only come to this PhD, otherwise don't come. There are several options present. You can use any of them, opt any of them. And this is very important. How mentor is helping the student to come from the comfort zone to the academic success zone. So comfort zone students feel safe and controlled. So nobody wants to go out in the sun when they are sitting in the AC room. But you have to, if you want to get academic success, you have to leave the comfort zone and come to the fear zone where they feel that they are lack of confidence, self-confidence, find excuses, be affected by other opinion. So the friend says that why you want to go there? You can spend some time with us and then we can go there, but not. You have to overcome this fear zone. Otherwise, you cannot reach their academic success. Then the learning zone comes, deals with challenges and problems. Because if you want to do something new, you have to face the challenge. Acquire new skill. You have to develop new skill. Because as I said that when I was a PhD student, nothing was explored with nowadays what my students are doing for all high advanced molecular biology training. So everybody has to learn the new skill to know something new. Extend your comfort zone. And finally, the growth zone, find the purpose, pursue the dream, set the new goals and conquer the objective. So if somebody wants to achieve the academic success, they have to cross all the 3 barriers. So that's why I said that mentors should provide opportunity and appropriate platform, environment to the mentee student to reach the growth zone from the comfort zone. So here the mentor has to play an important role so that he can mold the student to come to the academic success from the comfort zone. Now in our academics, there is only 1 parameter or 2 parameters to judge anybody's credibility, because until unless you publish or you do patent or develop some method, you cannot justify your credential. So there is a model transforming publish to perish model, but I made it publish to flourish model. So when a student comes for PhD, his main aim is to publish. And then he thinks, I tell that you publish and flourish. Publish in high impact journal and flourish. Publish frequently in high impact journal and flourish. And in that case, leadership and mentorship is very important, I can tell. And that is the reason myself and one of my students, Ashish, has written an article in Science and Society page of Trends in Genetics, Research and Leader a Twisted Tale, where we advocate that leadership is very important and that decide the fate of the student. If the leader is good, if the mentor is good, the student will get success. And once the student gets success, the leader or the mentor always get the reward. So I said that leadership is service, not a position. And that is the reason I have written the success of the mentee is the success of the mentor. So when the student gets the success, like I have shown, all my students, they got awards and I got the reward, the success also the mentor. And this should be realized by all the mentors. So thank you very much. This is my dedicated team. I think most of them are now left. Very few are there. And I have talked about Charu and talked about Roshan's, the second student and the last PhD student. And the middle one is Muthu, and funding from Department of Biotechnology and DST, Department of Science and Technology. Thank you very much.
Thank you, sir, for an excellent presentation. You have provided in-depth scientific evidence-based understanding of the importance of millets, how millets can enhance the nutritional security, and how it can feed the 10 billion by 2050 a genomics perspective. No doubt, millets is a wonderful crop. And besides this, also you have shown that how from the comfort zone to academic success, I mean, this part was also very interesting. And thank you so much, sir. Now I request Dr. Parmanand Barman for the question and answer session. Over to you, Dr. Barman.
Yes. Thank you, ma'am. Am I audible?
Yes, yes.
Yes. Yeah. Firstly, sir, we feel very privileged to have you among us. So being a for us, like plant biologists particularly, you are a role model for all of us. So we knew about your research, but I had no idea that you were such a good science communicator. So your lecture was a complete package. So like interesting example from Bollywood, from your experiences in food stall. It has traditional knowledge. You included gazette notifications. You covered nutritional aspects, economic aspects, environmental aspects, societal aspects. So it's like a complete from all. So our institute is a communication institute and policy research institute, but we also have research students. I know that our students will learn a lot from today's lecture, how to prepare a good presentation. So I think it will be very helpful for them. You had a good message for the PIs also. Ideally, the scientific presentations only include research outcomes, and then they are not like they are useful for a particular group of people only. But your lecture was complete from all aspects, and it will benefit all of us in some ways. So thank you so much for your lecture. And then it was a good learning experience for me as well. So before, we have our audiences. Please type your questions in the chat box if you have any question, or you can raise your hands for your question. So before going to their questions, so I have two questions, sir. You started with some examples of underutilized or neglected crops like Dioscorea and like other tuber crops. So we all know that International Year of Millet has revolutionized the importance of the attention of millets from the research community as well as public also. Like recently in G20, they were using millets in different forms. So how we can popularize the other underutilized crops that way? What can be done according to your opinion?
So I think like millets this year, a few years back, FAO has also dedicated the International Year of Legumes actually. So similarly for tubers and other United Nations and FAO should have taken this initiative to popularize the other underutilized crop species which are covered by tubers and legumes is already done and millets is already done. So I think tubers is and leafy vegetables is another two are left actually.
Yeah, thank you, sir. So another question is what measures can be taken to promote millet cultivation and consumption in India from audiences?
Which crops we need to explore more or to lower and cost and cultivation? I think all the millets are these because they are very hardy crop. Doesn't require any attention. You spread it in a very semi hard soil where no other crops can grow, but millet can grow without any taking water and fertilizer. Very less attention is required for any millet actually.
So we have a question from Rais, sir, I think. So please unmute and ask your question.
Hello, Professor Prasad. I'm Rais. I'm from Science Communication Division of NISPR, CSI NISPR. So I have a question. First of all, I think you're lucky. You have a wonderful group of students who actually have a lot of achievements. So,
look, Dr. Rais,
yes.
I have taken all students from all remote zones. Charu came from Jharkhand. Uttu came from Theni Valley, from Tamil Nadu. Theni Valley, nobody knows, very small village actually.
Right.
And
so they're all
Ashish
wonderful actually,
from,
as I could see.
Kashyong, he never holds a pipette. So
Right.
these are all dedicated people and they made their life by themselves. I have very little help. I just facilitate them to make their life easy actually.
No, I could also see the democratic approach. Actually, you are actually practicing. That's very well. I quite appreciate you actually. In fact, still I have a question regarding in your one of your slides you actually mentioned your long-term goal is design cereal crops which are more durable in adverse climatic conditions. So I have just a small question: how do you initiate this designing process? How do you design? In fact, how do you decide the particular crop I'm going to actually pursue with the genetic modification or whatever?
So in our institute, we are working different crops actually. So say for example, I work on foxtail millet. Few people work on the chickpea. There you go. And one scientist work on Navin-based work on brassica. Okay. So our goal is to develop some crop variety which withstand this adverse climatic condition and nutritionally superior. And for that reason, we use genome editing because this genome editing tool recently has been regularized by the government to release the genome edited crops. And for that reason, brassica, low uric acid, and that lines has been developed by Naveen Chandra Brist in NIPGR. That will give a classical example how modern biotechnical tools can be utilized to develop these durable high-yielding varieties in the current situation. Because what I have shown you, that overexpression, that was still a debate in the policy issue for, and Professor Pentel's Dhara mustard hybrid, THR11, government has regularized this thing, and the question is in the case is in Supreme Court. And I think over the time, few traits that has to be taken care, like BT brinjal, BT cotton. Okay, these are the two success stories that already scientists have shown, and most of the, like in Bangladesh and other Asian countries, they are growing this BT brinjal. But still, unfortunately, we are not in India. But most of the farmers has that BT brinjal seeds from percolated from Bangladesh. So what are the big variety, big size fruits we are seeing now? Even in maize, your corn, you are taking this sweet corn. These are all are transgenic actually, basically. In oil also, we are eating this canola oil. These are all transgenic actually, basically. So when our scientists produce, that comes under the regulation. But when we import from outside, we happily use it. So there is a bad policy issue. But anyway, we are being a scientist, our job is to tell that how we can better improve the varieties at the current scenario. And for that reason, your question is very pertinent. And genome editing is one of the tools that people can utilize. And this can be utilized by the current scientists. And we can develop some climate resilient crop by using the genome editing tools.
Thank you.
Thank you, sir. Mona Lisa Paul, please unmute and ask your question.
Yeah, thank you, sir, for such a thought-provoking lecture. It was indeed a very good one. So my question is, like, I have been on wheat and rice diet for the last 38 years. So suddenly if I switch on to this millet diet, won't it affect my alimentary canal or the digestion or something like that? Because millets are very heavy, they are very heavy cereals, and so they can cause cause diarrhea, not diarrhea, just loose motions and all. So what will be the take on that? Like suddenly, like I'm urging people to go for millets. So a person who was on a diet, a particular diet, wheat and rice for so many years, and suddenly changes to that diet, won't it affect the complete gastrointestinal process, sir? Your take on this topic, sir?
My take is this: we consume food for 3 times, breakfast, lunch, and dinner. I'm not telling you take millet for all the 3 times. You take millet sprout in the morning. You can take sometimes millet khichdi, like in foxtail millet khichdi was very good. You can take also sometimes the millet dosa. So once in your 3 times, why you can consume millet? That will not harm anything, and it will improve your, because millets are very rich in fiber, and also also all the nutritional I have shown. So don't take millet for 3 times. Take at least 2 times.
Okay. Thank you, sir. Thank you so much, sir.
Thank you. So we have two questions about the millet prices. Like as we see in market, the prices are very high. So they want to know about that.
As I said, the production area was very less and the production is also less. Now, once the European, they use this millet for birds' feed, now they are consuming, man. So that is the problem. So farmers, those who are producing, they are exporting this millet to the European and American countries because they are highly nutritious and the people are consuming the millet that cookies actually, or the for breakfast. Tata has Soulful has very good actually that millet, finger millet from the like in the breakfast we eat now with what with milk. So ragi from finger millet, they have produced a lot of this breakfast meal also from this, and cookies is also there. So that is the reason that the production is less, demand is more, and that is the reason the price is more than 100 rupees per kg for any millets.
Thank you, sir. We have one question from Biva. I think Biva, please unmute and ask your question.
Sir, good afternoon. So I'm Vibha from Science Media Communication Cell, NISpR. So I have a small question. And first of all, sir, your presentation and talk was very informative, I would like to say. So I have a small question. Sir, do you think millets has the capacity to replace other staple crops like rice and wheat in future?
Look, that's why that video I have shown by FAO that please bring me to your table. So why millet is telling? Because for the betterment of the all human civilization in the current situation, we need to get nutritious food. Food we are getting, no doubt we are eating food, but how nutritious they are, that is important. Because if you take anything, but if your body doesn't absorb this thing actually, then you have to take the supplement like iron supplement, calcium supplement. So, but if you eat this millet, I think there's no need to take any supplement.
So one thing I just want to know, does it have the capacity to replace all other cereals? Because we eat to satiate our hunger as well. So do you think it will replace all other cereals that we eat till now? And it's
again, this is a policy issue because you know that the area of cultivation is less, the price is very high, 100 rupees. Who is going to buy this 100 rupees? That's why when Professor Swaminathan said that it's a poor man's diet, now it is not a poor man's diet, it's a rich man's diet, man. Those who have money, I also buy it for my consumption. But I don't think normal people, those who have living in a very lower middle class, they can buy millet for their consumption because it's a high price. The dosa, more than 100 rupees. When you get within 50 rupees dosa anywhere in the market. But I think it will take time. People will realize we need to produce more millet and we have to popularize this thing, and we have to, because changing food habit is not an easy task. If I tell my student that I will make you a pancake with that foxtail, it is already available in the supermarket. Pancakes for all the millets actually, based pancakes, and banana pancakes is also there. Similarly, that millet based pancake are also there. So I told my 2 times I have brought from the supermarket and I have made it and put some chocolate on it. So my son ate it very easily. Because chocolate was there, so he cannot feel the taste of millet actually. But by this way, we can slowly bring it to our food table so that we can consume at least once in a while. Like in the broth, you were eating the kuttu atta. What is kuttu atta? This is a wild wheat, buckwheat. Again, is a cereal, C4, buckwheat. Okay.
Yeah. Okay. Thank you, sir.
And if you have a allergy with gluten, you can take easily this gluten-free. All the millets are gluten-free. Yes. Because I have seen several people have allergy in gluten.
Wheat protein. Gluten is a wheat protein.
Exactly.
Yes, sir. Thank you, sir.
Yeah. Thank you. So whenever we have some good presentations, it is normally followed by lots of questions. So if sir permits, I'd like to take 3-4 more questions.
Yeah. Please go ahead.
Sure. Thank you, sir. So Dr. Kanika Malik from CSR NISPR, ma'am has asked, how do you see millet cultivation in line with Green Revolution, and why wheat crop was chosen one for Green Revolution and not the millets?
Because you know that this is again the policy question, because the government has to, like promoting this millet here, the government has also tried to make some given to the farmers the incentive for millet cultivation, because no farmer is going to change the normal practice, rice, wheat, and any legumes. Okay, this is the normal practice. But if government gives some subsidy, then give that security that we'll buy the millet from you and then we can put it into the food chain, then I think a farmer will take it for cultivation. Otherwise, they're cultivating now also, but they're cultivating for their own purpose and for the limited use purpose. But once this government, through this policy, they can provide some subsidy, I think our farmers will definitely take it up, because they're getting a high-end value now for this selling these millets actually. If it is 100 rupees per kg, so it means that at least 70-80 rupees they will get.
So here I'd like to interrupt. I think the question was something else, what Dr. Malik has asked. She wanted to know that why during the Green Revolution, wheat was like the promoted crop rather than millets. Why millet was like not introduced for the Green Revolution that happened way back in 1960s.
Yeah, because that is again a policy decision, because at that time Norman Borlaug has came and visit India and gave some gift, that dwarf gene wheat and the rice also. And that is the reason that breeders has used this to cross it to our Indian varieties and introduce that dwarfing gene in our Indian cultivars. And by this way it happened, wonder happened. But again, I am telling you now that again policy issue. So if government tries put it in a positive way, so we can farmer will take it actually. Not like this that only wheat can be practiced, cultivated over the year and years.
How about introducing this in noon meal schemes in the schools?
Yes, again, I told you that this is a policy issue. The government has to, because I think in certain part in Tamil Nadu, I think it is a practice. Now, Tamil Nadu government is very proactive with using millets for their midday meal of the students, kids in the school. I think Tamil Nadu government is doing a fantastic job in that aspect. Even the area production I have found from this TNU literature, that the millet cultivation was area-wise increased in the Tamil Nadu state. If you take the last 5 years.
Thank you, sir. Ravi has asked how millets tolerate high temperature as compared to other crops.
So my whole talk was there actually.
Yeah.
You know that this HSP gene is only C4 crop harboring, no, not in C3. And when we introduce this foxtail small hits of protein, it can also grow, rice plant also grow at 50 degree.
So instead of changing the food habits, I mean, instead of it's hard to change the food habit, then change the crop accordingly.
Exactly.
So Dr. Charu ma'am has suggested one question. Any molecular breeding program initiated to integrate cis HSP 27 in foxtail millet or any related cereal crop?
No, I think we have to work on that aspect, because the breeding using foxtail is very tough actually. And what I have learned from this previous literature, that breeding is not easy in any of the millets actually. And for that reason, but I have already developed two population. One is for drought tolerance, one is for salinity tolerance. But for heat tolerant, I think we have to take a try by taking this, introduce this foxtail HSP 27 to rice. But I think it is a cross between foxtail millet and rice actually. So that is a very challenging job.
Thank you, sir. BK Paliwal has one question about global hunger index, GHI. So basically he's asking why we are at 125 rank even after plenty of foods.
Sir, I wanted to know, thank you very much, Dr. Manoj Prasad ji, for wonderful presentation and eye-opening rank of Global Hunger Index. You know that what are the are left behind so that go back, and what can be, if we know that particular parameter, then we can improve upon and can create an action plan. Generally, it is not a happy state of affair that we are lagging behind our neighbors, and we are looking that even after having so much food and this thing, and our global hunger index rank is so bad.
So again, I can tell you that we are sitting in an urban state. In city, we do not understand what is going on all over the other remote areas actually. So I think the data what is presented by the international agencies, it's very actually. Sometimes I do not want to believe this. But there is no other way to unbelieve this also, because these are all the statistics they produce over the time, and not a single year, but this is over the time. And the data for this global hunger index for Indian context, it is deteriorating actually. If you see I have shown you in the graph that from last 5 years, it is we are going down and down so far as the GHI is concerned actually. But I have do not know what is the reason for that actually.
Secondly, Dr. Saab, I also want to understand that edible oils and fats are very rich energy source, and whether means 900 calorie it has, while the other food articles have very less calories. So whether it has an impact of as we are dependent on edible oil import, is it so?
No, because that we are importing a lot of edible oil. But what I have told you that recently from our institute, Dr. Navin Base group, they have developed this low glucosinate content and zero uric acid containing brassica canola oil through genome editing. And I think this is the game changer for the oil point of view.
Very happy to report that I had been associated with National Dairy Development Board for more than a decade.
You know this thing.
I worked in with that, and Sankar brand we have Dr. Paintal has been the leader in this, and we 00 variety we have produced. Yes, we want to know that. I will search this thing that whether it is serving the purpose and linkages with the global hunger index, or it is irrelevant with that.
But I cannot comment on that, the global hunger index, because this is the data what we have seen from the literature actually. But what is the reason for that? It's very difficult to tell from a scientist point of view, from my point of view. But the data, whatever it is available in the literature, I have just presented. This is not a good sign for the country like us. So what is developing actually in a very good space actually. So
thank you very much, sir. It would be of great interest to know the details, what constitute the global hunger index, and what is that particular parameter which is lagging us behind, even after having so much good feel. But yes, we'll have to research and go deep down further. Thank you very much for your research and such a eye-opening presentation, and thank you very much from my side.
Thank you.
Yeah. I thank everyone for this engaging question and answer session. This was also very interesting. So if any of you have any more questions, please pass it on to me or Dr. Charulata, and we'll get the response from sir, and then we'll share the response with you. So thank you, everyone. Thank you, sir.
Thank you, Dr. Varman. It was very interesting, the question and answer series. Now I request Dr. Varman for the glimpse of SVASTIK activities on millets in partnership with MyGov.
So thank you, ma'am. So I'd like to inform the audiences that in our SVASTIK program, we have also collaborated with the MyGov platform. And previously we had organized a discussion activity, quiz competition, photography contest, and we received very good response across India. So then this year we have started some activities with the millets. So we have a short video where we'll show you a short glimpse of our ongoing activities and on millets. Computer. Stop.
Is there any issue or shall I share?
No, ma'am, we are loading it.
Okay.
Save update. Activity for chaos. Good.
Parmanand, is there any issue? I also have the video.
It's taking some time to load, ma'am.
Okay. Okay.
Is it visible, ma'am?
Yes, it is visible.
Yes.
Thank you, Dr. Varman. Is there some more videos to play?
No, this one only. So basically, we have received recipes across India, which we'll share in details with the audiences. And then we have one ongoing activity as shown in the video, which is on traditional ways of processing millets. So I'd request everyone to participate in that activity if you know about any traditional ways of processing millets. Thank you.
Thank you, Dr. Varman. Now I request Dr. Charulata for the vote of thanks.
Thank you, Dr. Suman. So yeah, So it was indeed a very, very nice lecture, and I deem it a great honor and privilege to propose the vote of thanks on behalf of CSI NISPR and Team Swastik for today's Swastik lecture session. So millets were the first crops and millets are the future crops, and what better occasion than World Food Day to celebrate and promote these superfoods. Here I would also like to inform our audience that through Swastik we have also shared and disseminated a story on these traditional crops, millets. And we have completed one activity on MyGov. We have already shown one is live, and we are in future also planning one more activity on millet-based activity with MyGov. And for this Swastik lecture session, first and foremost, I'd like to thank our keynote speaker, Dr. Manoj Prasad, a renowned academician and researcher who has extensively worked on foxtail millet, and who, despite his extremely busy schedule, agreed to my request, I'd say rather at a very, very short notice, to grace the occasion and deliver such an engaging keynote address on a very, very pertinent and apt topic to feed the 10 billion by 2050. And he, being a renowned genomicist, gave his view from the genomics perspective. And I'm sure the audience who have joined us through MS Teams platform or those who have joined us live through Swastik Facebook page must have thoroughly enjoyed his presentation and must have been also immensely benefited from his talk. I would like to mention here that Sir, being my PhD mentor, and the fact that he showed my work at this platform when I have invited him to deliver a talk at a place where I work. Actually, it had made me quite emotional when you were showing my work that I left around 12-13 years back. And this actually calls for an additional token of appreciation and gratitude from my side for giving me the opportunity to work on this traditional crop, foxtail millet, that I'd work, and also accepting my request to be here with us. I also express my heartfelt thanks to our director, Professor Ranjana Agarwal Madam, for her stewardship, support, vision, and commitment to promote Indian traditional knowledge and to promote Swastik. She has been a guiding force behind this initiative, and her words of encouragement, able and continued support for this program is deeply acknowledged. I am very, very grateful to Shri R.S. Jayasomu Sir, Chief Scientist, SCSI NISPR, for presiding over today's function in absence of Director Ma'am. And Sir has always been very kind and supportive towards all Swastik endeavors, and his unstinted support in organizing this lecture is duly acknowledged. I'd like to thank the IT team, especially Shri Pavanji, for helping us with the online meeting and live streaming of the lecture. Thanks are due to all admin and finance staff who have helped us in organizing the program. Special thanks to the organizing committee, especially Dr. Suman Ray for the moderation, and Dr. Burman for the question and answer session and showing the video, and also the Swastik team for the wonderful execution of this program. I thank all our colleagues at NISPR and guests and audience who have joined us through NISPR Swastik page, Facebook page, and at my at MS Teams platform for taking out time and attending this program. I thank the media members also for the coverage. Kindly pardon me if I've left out any name, but my heartfelt thanks to each and everyone for making this program a grand success. Thank you once again.
Thank you, sir. Thank you.
Thank you then. I may take leave.
Yes, sir. Thanks a lot for coming and accepting my invite. Yeah.
Thank you. Thank you.
And I think, and I'm sure that I'll be calling you in person also. This time we can do it. It was a very, very short notice.
Yeah. It's a nearby place.
Yeah. Yes.